Hello, my name is Allison Sitton, and today I will be talking about nonsense-mediated mRNA decay. First, I would like to cover the critical function of messenger RNA. It is related to the primary sequence of its protein coding region, and thus this is subject to inspection by the cell. Nonsense-mediated mRNA decay is a eukaryotic surveillance process that monitors cytoplasmic messenger RNA translation and selectively degrades messenger RNAs harboring premature termination codons. This particular pathway exists in all eukaryotes. From yeast to humans, activation of nonsense-mediated mRNA decay requires the function of the three conserved UPF factors, which are UPF1, UPF2, and UPF3. In particular, creation of a premature termination codon generally activates the nonsense-mediated mRNA decay pathway. Premature termination codons can be introduced as a result of genetic or somatic mutations, mistakes during transcription, or mistakes during splicing. So now I'm going to be going over the mechanism. This mechanism operates in the cytoplasm and is intimately linked to translation termination. For messenger RNAs, there are three basic steps. The first step is detection. This is when a defective polymer is first recognized. The second step is tagging, and this is when the polymer is subsequently earmarked for degradation. And the third step is destruction, and this is when the defective polymer is then destroyed. So now I'm going to be going over the steps in a little bit more detail. So we're going to start with step 1, which is detection. The detection step for nonsense-mediated mRNA decay relies on two signals. The first signal is provided by the premature termination codon, which generally defines nonsense-mediated mRNA decay substrates, and the proteins that associate with the translation termination complex that assembles at the premature termination codon, including the terminating ribosome and the SIRF complex. The SIRF complex consists of SMG1, UPF1, ERF1, and ERF3. The second signal often derives from an exon junction complex, which associates with messenger RNAs less than 24 nucleotides upstream of a splicing-generated exon-exon junction, whether or not they are nonsense-mediated mRNA decay substrates. Other signals, such as unusually long 3' UTRs, also exist. The exon junction complex is composed of four core components, which are IEF4A3, Y14, MAGOH, and BTZ, and associated nonsense-mediated mRNA decay factors such as UPF3 or UPF3X and UPF2. If a premature termination codon is located greater than let's say 50 to 55 nucleotides upstream of an exon-exon junction, then the messenger RNA is recognized as abnormal and then tagging proceeds. So this figure demonstrates detection. So here we have the premature termination codon, which is what signal 1 is. And then we have the SIRF complex, and here are the components of the SIRF complex. For signal 2, we have the exon junction complex, and here it is magnified, and these are the components that are in the exon junction complex that I just mentioned. And this signal happens only when the premature termination codons on a messenger RNA are more than 50 to 55 nucleotides upstream. So the next step is tagging. And for tagging during nonsense-mediated mRNA decay to occur, all or some of the SIRF complex joins the exon junction complex, possibly while the terminating ribosome is still present. So this forms a decay-inducing complex. This configuration activates SMG1 to phosphorylate UPF1. Phosphorylated UPF1 signals the messenger RNA for destruction and has the added effect of inducing translational repression of the messenger RNA, which is a prerequisite for destruction. So this figure demonstrates tagging, and if you remember from the previous figure, this right here is the exon junction complex, and this right here is the SIRF complex, When these two complexes come together, as you can see right here, this forms the decay-inducing complex. And this activates SMG1, which phosphorylates UPF1, which is also shown right here. And these are the phosphorylation tags that are located on this factor. So now I'm going to be talking about step 3, which is destruction. Nonsense-mediated mRNA decay-dependent destruction of messenger RNA relies on recruitment of SMG6 and or SMG5 SMG7 complex or SMG5 PNRC2 complex via the phosphatate tags on UPF1, which was shown in the previous figure. SMG6 possesses its own endonucleotic cleavage activity, and this cuts the messenger RNA target at the 5' to the exon junction complex. 3' to 5' exon nucleases degrade the 5' cleavage product. So now UPF1 helicase activity disassembles the ribonucleoprotein components that are bound to the 3' cleavage product, and this is followed by 5' to 3' exonucleotic degradation. So the SMG5 SMG7 complex or the SMG5 PNRC2 adapter complex direct exonucleotic degradation of the messenger RNA. These adapters recruit decapping enzymes and or deadenylation enzymes, and their activities are followed by 5' to 3' and also 3' to 5' exonucleotic decay. So this figure describes destruction, and this is the SMG5 complex, and this is the SMG6. And we're going to be talking about the SMG6 first. So what this does is it cuts right here and causes an endonucleotic cleavage, which gives us 3' to 5' exonuclease and also a 5' to 3' exonuclease. So now we're going to talk about the SMG5, SMG7 complex, or we could also talk about the SMG5 and the PNRC2 complex. What this does is this complex here adds a decapping enzyme and or a deadenylation enzyme, which gives us a product of 5' to 3' exonuclease and also a 3' to 5' exonuclease. So now I'm going to be talking about what happens after destruction. So after destruction of the messenger RNA, nonsense-mediated mRNA decay factors must be recycled for further rounds of decay. As discussed, UPF1 helicase activity disassembles components of the nonsense-mediated mRNA decay substrate for future use. UPF1 itself, however, must be returned to its basal hypophosphorylated state. And this is accomplished by protein phosphatase 2A, which is recruited by the SMG5 SMG7 complex. Dephosphorylation of UPF1 is critical because failure to recruit protein phosphatase 2A then leads to an inhibition of nonsense-mediated mRNA decay. Additionally, the ribosome must be recycled, but both the timing of the step with regard to the messenger RNA destruction and the identity of the factors contributing to it are unknown. So just to summarize everything that I've previously talked about in this presentation, nonsense-mediated mRNA decay is a process that monitors cytoplasmic messenger RNA translation, and then it selectively degrades messenger RNAs that are harboring premature termination codons. It requires the function of the 3 UPF factors, and it also has those 3 basic steps, which step 1 being detection, step 2 tagging, and step 3 destruction. So for the events after nonsense-mediated mRNA decay, the factors must be recycled to be used again. Then the ribosome and other factors are reused in the next round of decay. And this concludes my presentation. Thank you.